Uh, Good evening.
Good evening, madam.
Um, I have a concern with regards to the tuitions.
Yes.
We'll be I'm sure you're aware that we'll be going for our residentials.
Mm.
Yes, and it shows that we'll just be there for a week. So I don't know what others think, but from my experience last time we went, we hardly had time to have enough. We hardly had enough time to even study on our own. So I was just thinking, can we maybe postpone the tuitions until we're back from residentials? I don't know what others think, because I think it would be a challenge for us to join in, because sometimes you find other tutors who also request for online classes in case they missed what's this they missed a class. They would request, no, we should do online and all. I don't
Okay.
know what others think. Yes.
Okay. No. The suggestion is welcome. The suggestion is welcome, madam, and I'd love to hear from what others think as well. If your majority want that, there's no problem. We can pause a little bit after the extensions are done. That's when we resume. There's no problem.
Okay, sir.
Yeah.
Thank
Okay.
No,
So I
good
think
evening, sir.
good evening, madam.
Yes, for me, I was thinking that maybe we don't postpone, but at least we should be revising for all my topics that were done at school. That's what I'm thinking. I don't know what others think.
Revising.
Mm,
Okay.
yes.
Okay.
Good evening,
Okay.
sir.
That's good evening, madam. Yes, madam.
Me, I'm in the shoes of the first speaker because
Okay.
biochemistry is being taken by different faculties like us, general nurses, the midwives. So you find that on a day that midwives are having a maybe community diagnosis test, as we are having a different test that we have to prepare for. Since we've been told to say this for a week, will be dedicated to much of the tests and like learning. So I feel it will
Mm-hmm.
be a little bit difficult where we are revising anatomy. I have to prepare for maybe com skills tests. So I'm in the shoes of the first speaker.
Okay. 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 So that's what are you thinking?
Good evening, Doc.
Good evening, madam.
Yes, I think I also second the first speaker, considering the
Okay.
fact that we'll be knocking off at 18, and usually we like for last time we used to
Mm-hmm.
get very so, and plus the congestion in Lusaka. Some of us are coming from home, so the congestion for us to reach home it will be maybe 20 somewhere there. So if you say that no, we start revising, I think most of us won't manage. Yeah,
Okay. Okay.
but
Okay.
then we'll
Okay.
continue having anxious since it's
Sir,
just
good
1 week.
evening.
Yes,
Yeah,
madam.
I'm
Good
actually
evening.
in support of the first one,
First
just
one.
as
Okay. Okay.
yeah.
Okay, team. Then no problem. We shall do that. When are you starting the residentials?
Monday.
Monday. Oh, okay. This coming Monday. Okay, we
Yes.
shall do that. So it means it's only lasting for a week, right? And I understand I'm teaching different programs. So for all of you, like you're all going for residentials.
Yes.
Okay. So and then up to you said this usually takes 1 week.
Up to 10th of August.
Okay. There's no problem, team. I think we can pause and then what we'll do, yeah, you make payment like the way we do, and then next month I'll have a week that we're not going to learn. Yes, before making the for that other month. So there's no problem. We can do that.
Yeah.
Okay, 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 sure, no problem. So, which means we're going to have our last lesson on Sunday, and then after work 1 week, you let me know when we're done. Okay, so let's start, Tim. Let's talk about. We're looking at now. Okay, so what we're looking at the glands. Last time we discussed the epithelial cells, and the different type of epithelial cells, and also where we are going to find them. Yes. So now we're looking at the glands. So I want us to look at the glands, and then we can also look at the muscle tissue. Okay, so when you talk about glands, what are these glands, and what do they do? So you have to remember that glands, these are simply specialized epithelial structures, and they're made up of epithelial cells that we discussed last time. So the glands, these are simply specialized epithelial structures that synthesize. So they are synthesizing, okay, and secrete what? Substances. So we are producing and also secreting substances in these glands. Okay, so what substances are we producing? We are producing things like hormones. So hormones are produced in the glands. Things also like enzymes. Also producing enzymes. We're producing mucus. We're producing sweat, saliva. Okay, sebum. All those things are produced in the glands. So you have to remember that we have got different classifications of glands, okay? So we can classify glands. Number one, we have classification, okay? Classification based on the route of secretion. Based on the route, okay, of secretion. You have to remember this is our first classification, okay? So based on the route of secretion, what are these classifications that we have based on the route of secretion? So the number one thing, what we have is what we call exocrine glands. Now, what are exocrine glands? Because remember, we are classifying based on the route of secretion. How are we secreting things that we are producing? Let's say we produce hormones. These hormones, how are we secreting the hormones? Okay, that's how we are classifying them based on the route of secretion. So we have got what we call the exocrine. Now, what are these exocrine? So the exocrine glands, I want you to remember that these are glands that secrete their products through ducts. Okay, so these are glands. These are glands that secretes products through ducts. Through ducts. That's a key word. So in these glands, we've got ducts that are going to secrete what? That are going to secrete the product that we're producing. So for example, imagine this is a gland. This gland is going to be like this. Okay, it's made up of what? Epithelial cells. And then it has got a tube structure like this. This tube now, let's say, is going to secrete, let's say this is the stomach. Okay, so we are going to secrete where? Into the stomach. So this is what we call a duct. This one is a duct. Yeah, so you have to remember we've got the exocrine. They are producing here and then secreting through a duct. As simple as that. Okay, so what are some of the examples of exocrine? Examples that we can give, we've got so many examples of exocrine, but we can give one: sweat glands. Okay, sweat glands, the one that produces sweat. These are what are exocrine glands. They have got these little ducts. Okay, apart from that, we also have salivary glands. They also produce what? They're also producing through ducts. Apart from that, mammary glands, the one that produces milk. Mammary glands, also the same thing. Okay. Yes. So we have so many of these glands, but just want to remember. I want you to remember that the number one feature is what? Ducts. This is something you didn't forget. Now, if we have something that is secreting with a duct, we also have what we call the endocrine. So number two, we have got. So remember, the first one we said what? Exocrine. We also we have got endocrine. Now, what are endocrine glands? So just remember the keyword there. Exo means the outside. Okay? So it's secreting through the duct to the outside. Endo means inside. So endocrine glands, these are glands that lack ducts. Okay? So I want to remember that these are glands, just the opposite of exocrine, are glands that lack ducts. Okay? So if they lack ducts, they don't have these tubes. How do they secrete? So they secrete and they release hormones directly into bloodstream. Directly into bloodstream. So how is it going to look? So imagine, let's say I've got a gland like this. This is a gland, just going to be a little gland like that. And then here there's going to be a blood vessel. So it has no duct. Whatever it's producing here will just go into the blood vessel by diffusion. Okay? There is no any duct to secrete that. These are called endocrine. So you have to remember endocrine and exocrine. Now, what are some of the endocrine glands? Okay? What are some of the examples of endocrine? So examples. Okay, so examples. We have got things like what? Pituitary. So in the pituitary, you are going to find glands there. Okay, endocrine glands. So pituitary, we have got things like thyroid gland. Okay, all those are endocrine glands. Okay, so pituitary gland. So in the pituitary gland there, you're going to find what? You're going to find these endocrine, small glands. Yes, so endocrine produces what? We know that endocrine produces growth hormone, okay, thyroid stimulating hormone. We have got follicle stimulating hormone, luteinizing hormone, all those hormones are important to reproduction. Yes, so all those are produced in pituitary gland. So pituitary gland is one of classic examples. Apart from that, thyroid gland. We all know the thyroid gland producing thyroid hormones. This is also a what? It's also an endocrine gland. So this one produces T3 hormones, those thyroid hormones, and T4, calcitonin, all those are produced there. Another example I can give is what? Adrenal glands. So there are so many glands that fall under endocrine. Adrenal glands produces the cortisol. Okay, cortisol is also producing what? Aldosterone. Okay, so these are simply some of the glands. Okay, endocrine glands. Okay, so I want to remember that we have got exocrine, endocrine. We also have mixed. Okay, see, mixed glands. We also call them what? Heterocline glands. Now, what are these? To me, simply it has both exocrine and endocrine function. So, which means it has a duct side, and also one side it doesn't have a duct. Okay, so it's like it has got both features of the exocrine and endocrine. Okay, has both features of endocrine stroke exocrine. Okay, so what example can we give? The pancreas. Okay, so the pancreas, what does it mean? Pancreas, in the pancreas, you are going to find exocrine glands. Apart from that, you are also going to find endocrine glands in the pancreas. Okay, so pancreas, we know, produces insulin, glucagon. So it has both. Some of the cells, some glands there, they are exocrine glands. Some glands are endocrine. Yes. Apart from that, ovaries. In the ovaries, the same thing. Okay. In the testes, it's also the same thing. There's going to be what here? There's going to be double. Okay. So remember, if they ask you what's the classification based on the route of secretion, you say exocrine, endocrine, and mixed. Okay. Now, we can also classify glands based on the number of cells. Okay. So classification. Okay. Based on the number of cells. Okay. Now, what is the classification based on the number of cells? So I want you to remember that we have got unicellular and multicellular. Okay, very simple. So we have got unicellular. Okay, and we also have what? Multicellular gland. So what does it mean? Unicellular, it simply means it's just simply one cell. So imagine this one cell like this. And this cell, it even secretes what? Secretes enzymes, or maybe it's secreting, let's say, hormones, or some mucus maybe. It's just one cell. That's what simply means. Uni means one. Multi means a lot, multiple cells. So unicellular is simply one cell, okay, that is producing substances. What example can we give? A good example is a goblet cells. The goblet. Goblet cells, very important. You're going to find them in the respiratory tract. You also find them in the small intestine, large intestine. You find the goblet cells. What do? They produce mucus. It's simply one cell. That's how the goblet cell look like. It's just like one cell. Yes. So that is unicellular gland. Then multicellular now, it means now these glands have got what? Multiple cells. Okay? Multiple cells. Okay. So all these glands now, all these glands that we know, salivary glands, sweat glands, they all fall under multicellular. It means they're made up of many cells. Okay, that's what you have to remember. So you can classify based on the route of secretion and also based on the number of cells. Okay, so now after classification that one, I also want you to know that we have got what? These exocrine glands. Okay, we can classify them further. So these exocrine glands, we can classify them further. We have got different types of exocrine glands. So let me just draw that. Let me just add an image. Just trying to add an image so that you can see that properly. Okay, so I'm sure you can see this. Okay. So these are simply the glands we're talking about. So you can see that we have got the unicellular gland. Okay, this is simply like one cell. Yes, for example, the goblet cells. So these are called unicellular. So we are going to find like I said, in the respiratory tract, in the trachea, bronchi, you find the goblet cells, in the small intestine as well. Okay. Now the other, so these are classification of what? These are, so if you can see here, these are what? Exocrine. These are exocrine glands. What does the exocrine glands are? They have got a tube. This is a duct. This here that you can see. This tube-like structure. Oh, this is a duct. This is a duct. So these, they have got ducts. So we call them, we are calling them what? Exocrine glands. So these exocrine glands, as you can see, they are different types, just based on the structure, how it's looking. So you can see this one here, it's called a simple straight tubular gland. Yes. So what do you know? Just a one tube. So this one is called simple straight tubular gland. Okay. Where can you find it? You can find these glands in the intestine. You can find the intestine, you can find it in the colon as well. You are going to find this. Okay. So yeah, I want you to remember that when you see a straight tube like this, because sometimes they can ask you maybe in the exam, they just draw for you this. And they ask you, label, what type of glands are these? Those are kind of questions you can expect. So usually in the labeling, they give you a gland like this and without a name, what is this gland? I want you to remember from this lesson that just looking at the structure, it is straight, okay? And it has no branches, and it has a duct. So we call this one simple straight tubular gland. Okay. Then we look at this one. How is it looking? It has branches and it has a tube as well. This is also called simple branched tubular glandine. So this one is straight. The only difference is that this is simple straight, this is simple branched. Okay, now where can you find this? This one, you're going to find it what? In the stomach. Okay, mainly in the pylora sign. It's an area of the stomach. So you find it in the stomach. Okay, and you can also find it in the intestines as well. Okay, so this one mainly secretes mucus. These are mucus glands. So remember, in the stomach, this one also will be in the stomach as well and in the colon. Okay, so just remember this is simple branched tubular. Then we look at this one. How is it looking? It's looking coiled. So how do we call it? Simple coiled tubular. Okay, so number one, the way we are naming these, I'm sure you can see the pattern. We're looking at what? The shape. Okay, what is the shape? What is the structure? Okay, so remember this is simple coiled tubular. So this simple coiled tubular, where can we find it? We also find it in the pylorus. Okay, and also in the small intestine. This one, what is it doing? Mucus secretion as well. Then the other one that we have is what we call the acina. So this acina, how is it looking? So the simple acina, we also call it what? The simple alveolar. That's the other name, So we can also call it simple alveolar. Looks like the alveolar of the lungs, glands. So what is this? Where can you find it? So this one, you're only going to find it what? In the skin. Okay? You find it in the skin. So these are the sebaceous glands of skin. Yes, produces what? Sebum. Sebum is like oil that lubricates skin. So sebum, this is simply the oil that is produced and lubricates skin. So you're going to find this in the skin. So when you see the structure, this is a simple acina. Okay. Then, apart from that, so remember, when we talk about glands, we've got the simple and the compound. Okay. So the simple, they have got basic structures. The simple have got basic structures. They don't have a lot of branches. Those are called simple. And then the compound, they have got what? So many branches, as you can see. Okay? So remember, compound glands have a lot of branches. And then simple, they have just few branches. Okay, so let's talk about this one, the simple branched acina. The simple branched acina, as you can see, okay, it's also found where? You also find it in the skin. Yes, so this is also producing sebum. Okay, simple branched acina. So you're going to see a lot of these. These round structures, we call them what? Acini. Okay? So these are called the acini. One is called acinus. This round structure you can see there. And then this is a tube. Okay? So these we call them simple branched acina. Most of them are found with what? Are found with hair follicles. They're found in the skin. Produces sebum as well. Next one, these are the compounds now. These are called compound. Look at the branches. So just remember, if there's few branches, like the way you can see, it's called simple. If there's a lot of branches, we call it compound. So there are two, compound and simple. Okay? So the first one is compound tubular gland. This one, it has got a lot of branches and it has a tube. Why? Yes. So just remember the key word. If you see these round structures, the name will end up with acina. Okay? If it's a tube, the name will be tubular. Okay? So you can, even if let's say you forget, you just said a gland that's looking like this. Okay? Few branches, maybe something like that. what can come in your mind? In your mind, you're going to think of what? You're going to think of a simple asinine. Yes. Yes. So simple branched acina. Why? Because it has got what? Some few branches and then also these acinas. Okay, in plural we call them acini. So just look at it. Is it simple? Simple, it means that there's few branches. Two, what else am I looking at? I'm looking at the glands here. How are the glands? Are they round? If they are round, I'm going to call it what? Acina. Okay? So I'm going to call this what? Simple, branched acina. Okay, if it's just like this, that is simply what? My simple acina. No branch. Okay? So that's one thing you have to remember. Then let's look at this compound. So if you, I'm sure you can see here, these, they are no round structures. It's just a tube throughout, so we call them tubular. So the first one, like I said, is a compound tubular. This one, you are going to find it where? An example I can give, you find it in the duodenum. Okay? In the duodenum, there's what we call Brunner's gland. They're found in the duodenum. Brunner's gland, this is a secrete mucus, okay? Yes, so these are found in the duodenum. The Brunner's gland is a good example of compound tubular. They look like this, those glands, okay? So they produce what? Secrete alkaline mucus to neutralize the acidic stomach content that's going into the duodenum. Then the other one that we have is the compound acina. Compound acina, I'm sure you can see, okay? This is a compound acina. And this compound acina, what do you have to know about it? So you can see it has got these branches, okay? And it has got these acini. So what example can we give? A good example can give is the mammary glands. Mammary glands, okay? The one that produces milk in the breast. Mammary glands, that's how they look, okay? So remember, mammary glands that produces milk. Even also, some glands in the pancreas as well. They are also looking like this, especially those that produces enzymes. They look like this. The last one is this one that you can see, the compound tubuloacina. Okay, compound tubuloacina. So this one, it means it contains both. Okay, tubular and the acina. Excretory portion 9. Yes. So if it's containing both, we call it compound tubuloacina. You find this mainly in the pancreas. Apart from that, you also find it where? Salivary glands. Salivary glands, they look like this. Salivary glands. Okay? So remember pancreas, you find it there, and those salivary glands. That produces saliva. So I want you to remember those types, team. These are the Okay, so these are the types of glands that I want you to know. And so these are types of what? Exocrine. Types of who? Exocrine glands. So please remember the classification that I gave you and also how we classify the exocrine glands. So I want you to remember that. That's very important. I don't know if there's any question. Okay, now just the last one, 9. The last one thing. Yes. The last one thing I wanted to talk about is the classification of these glands. Okay, classification based on the mode of secretion. Remember, first one I said was based on the route of secretion. Now it's based on the modes. How do we produce the secretion? Okay. So we have got 3 classifications, and this one they like bringing it in the exam. A, we are going to say what? Merocrine. So they are merocrine glands. Okay, so it's what we call merocrine secretion. So gland that secretes through what we call merocrine secretion, we call them merocrine glands. B, we have what we call apocrine secretion. Glands that secreted by apocrine secretion, we call them apocrine glands. 3, or C, we're going to call it holocrine secretion. Now, what is this secretion? What am I talking about? So when you talk about merocrine, merocrine, what's happening is that a molecule is going to be secreted. Imagine this is a gland. The gland has got cells. Now let's imagine that this is a gland. Okay, this is a gland like that, and then these are cells. Remember, we've got epithelial cells in the glands like that. So these are cells. Now let's imagine that we get one cell and look at it. So this cell, when it produces a content, okay, let's say it produces enzymes. Enzymes are proteins. I'm sure the other day we discussed protein synthesis. When enzymes are these proteins are produced, they undergo what? Post-translation modification, where those proteins are going to add phosphate, all those things. Now, after you do that, these proteins, they go to where? To the endoplasmic reticulum. So from ribosome, proteins go to the endoplasmic reticulum here. This one, it's going to pack these proteins, okay? And take them to the Golgi apparatus. The Golgi apparatus, so imagine these are proteins. When they go to the Golgi apparatus, this Golgi apparatus, it puts these proteins in a what? In a sac. This sac, we call them vesicles. So imagine, let's say, like you put proteins in a balloon, okay? That balloon, we call it vesicles. Now, these proteins, once they are put, so imagine this is an enzyme. I mean, this is an enzyme that we have produced. This is the protein I'm referring to. So this protein or this enzyme, once you make it like that, you put in a vesicle, you are going to release it. Okay? Now, when we release it, there's a process that we call exocytosis. Okay? So what is exocytosis? Exocytosis, it's simply where this vesicle, it will come here. Okay? When it comes there, it's going to fuse with the cell membrane. So imagine, let's say this is it has come there, this is the cell again, okay? And then in this vesicle, it will come and fuse, join with the cell membrane, so that now it looks like this. Have you seen? So this vesicle joins with the cell membrane, and then the cell membrane will open up this side. And then we are going to release what? Our content. This is called exocytosis. Okay, so remember, if a cell is releasing its products through exocytosis, that is what we are calling merocrine secretion. Okay, so which means that we are not damaging the cytoplasm. This one, the vesicle, will come and join with the cell membrane, and then we release our products. Cytoplasm, the cell membrane will remain what? Intact. Yes, that's what you are calling what? Exocytosis. So when you hear merocrine, just think of what? Exocytosis. Exocytosis. So glands that produces or that releases its products through exocytosis, we call them what? Merocrine glands. Okay. Now, what is an example of merocrine glands? Salivary glands. Salivary glands, this is what they do, what I've just explained here. That's how they release when they produce saliva. Okay, salivary enzymes, all those things. Okay, salivary amylase, those enzymes. Once they are produced, they are released the same way I've explained. So salivary gland is one example. Okay. Apart from that, even sweat glands is also another example, and they also do the same thing. Now, there's also what we call now apocrine secretion. Okay, what is apocrine secretion? Apocrine secretion now, here I want you to know that what's going to happen in apocrine secretion is that the apical part of a cell is going to pinch off with the secretions. So what does it mean? what it means is this thing. Imagine this vesicle when it comes, it will join there. It's going to join the cytoplasm and the cell membrane there. Then this part of the cell membrane will pinch off together, will come out together with the products. So this part of the cell membrane, it's going to cut off, okay, and be released together with the products. So remember, in apocrine secretion, the portion, okay, the apical portion of the cell is going to pinch off with the secretion. Here, what was happening in exocytosis, I said there was no damage to the cell membrane. But here, in this one, part of the cell membrane comes out as well, is cut and released together with the secretion. Okay, so I'm just wanting to say that apical portion of cell pinches off. This is what apocrine. Now, which glands are you going to find? Which gland are you going to find this apocrine? You're going to find it where? Mammary glands. Okay. Mammary glands, and you find that. The last one is what we call holocrine. So the holocrine is just what? The whole cell will rupture. Okay. And become the secretion. So imagine, let's say you want to secrete this, you produce this. This whole cell is going to get damaged and rupture, and everything is released. That's how some glands do. So all the cell will just break down. So entire cell rupture. Okay. And become secretion. What is an example? Sebaceous glands. So please you have to remember this sign, what it means. I just wanted to add on that information. Okay, so that is quite important. I don't know if there's any question on glands, otherwise that's all you're supposed to know on glands. So what I want to emphasize on glands, just know what is an exocline, what is an endocline, what is a mixed line. Apart from that, make sure you know the types of exocrine, these ones here, and how they look. Okay, and also you have to know merocrine, apocrine, holocrine. And at least once you know these, you're supposed at least to be able to at least even give an example, one example of each. That's important. Okay. Yes. So let's go to anatomy. Okay. Okay, just a moment. Let me just add some notes. Okay. Yeah, so we are looking at now muscle tissue. Yes, anatomy. Okay. So I'm sure you can see a little mouse there. Now, my question is, what's the relationship mouse and muscle? Okay. So when you look at the word the muscle, actually the word the muscle, it comes from what? A Latin word, musculus. Musculus simply means what? Little muscle. Okay. So this came from Romans, ancient Romans. So actually these people, they used to think that biceps, they used to look like a little mouse. I'm sure you are able to see if you flex your biceps, it looks like a little mouse and moving. Yeah, so that's how they named these muscles musculus. So remember, muscle word come from Latin word musculus, which simply means the little mouse. Okay, I'm sure you can see there. And then there are also some other terms we have to be familiar with, like for example, sarco. Sarco means flesh. You're going to find that most when we talk about muscles, we use sarcoplasmic reticulum, okay, sarcolemma. We use sarco, okay, as a prefix. So this sarco prefix, it comes from it's a Greek word which simply means flesh. Okay. So, I want us to talk about the muscles. You have to remember that this muscle tissue is derived from what? The mesoderm. I'm sure you can see that here we have got, this is the muscle fiber that you can see there. So muscle fiber, this is the basic unit of muscle tissues, okay? Now, there are types of muscle tissues. There are actually 4 types of muscle tissues. Yes. Now, these types, you have to remember that, we have got 3 types. We have got what we call cardiac, smooth, and also skeletal. These ones that you can see, these are simply 4 types of tissues. We've already covered connective tissue, epithelial tissue. So today we are on the muscle tissue. So remember there are 3 types of muscles. So there's what we call cardiac, skeletal, and the smooth. Okay. So if you look at here, this is simply showing us the structure, but I just want to explain something on the types of muscles. Okay. Let me just add the page. And before we talk about the types, what are the functions? So the functions, those are simple things I'm sure you can see. The functions of muscles, body movement, maintenance of posture, helps us in respiration. Remember the ribs. Okay, we've got the intercostal muscles, which helps us in respiration. Okay, production of heat. When you're shivering, the reason we are shivering is to produce heat. Okay, for communication, we know we're able to, when we're talking, constriction of organs and vessels. Remember the blood vessels are made up of muscles. Okay, for heartbeat. So there's so many functions of muscles. Okay. Now, my question is, what are the types? Like I said, this is cardiac, skeletal, and smooth. Now, what are the differences? That's very important that you have to know. Okay. What's the difference between these? Okay. But before I talk about the difference, you have to know also the properties of muscles. What are the properties? This is very important. Okay. So we have got the excitability. What does it mean? It means that when you give a stimulus to a muscle is able to respond. For example, let's say electrical impulse. A nerve sends electrical impulse to a muscle, that's a stimulus, a muscle is going to respond. Okay? Even electricity, muscle respond to electricity. So you have to remember excitability. 2, contractility. The muscle is able to shorten with force. That's what it simply means. And then extensibility, it means that the muscle can be stretched, okay, to its normal resting length and beyond to a limited degree. Okay, so it means it's able to extend in short time. Muscles like this, when you pull, it's going to extend. The other one is elasticity. So it means that when you extend the muscle, it has to come back to the original length. Okay, that's what. So these are the properties. Please remember these properties. Very important. Now, like I said, these are types of muscles. Now, what is the difference? Okay, you have to know that, Tim. What is the difference between these types of muscles? Very, very important. These are questions that you can find. They don't miss, especially in anatomy on muscles. Okay. So when you talk about skeletal, okay, when you talk about skeletal muscles, okay. So imagine you've got skeletal muscles. Apart from that, we also have the cardiac muscles and the smooth muscles. Now, what should you know about these types? I want you to know that you can start with the control. Okay? What does it mean? These skeletal muscles are voluntary. Now, what is voluntary? It simply means that for them to work, supposed to do what? To stimulate them as a person. Yes, you're supposed to stimulate them. That's why, for example, if you want to take a walk, you're supposed to think, okay, let me walk. When you think like that, you send what? Electrical impulse to these muscles. So supposed to think about it. Okay, that's what it means voluntary. Without you moving them, they can't move. But when you talk about these ones, the cardiac, these are involuntary. There's no time when you are going to say, okay, today I don't want my heart to beat. The heart is beating every single time. You can't control the heartbeat. Okay? Yes, what it simply means. Even the smooth muscles, these are found in the what? You find them in the blood vessels mainly. Okay? Even in organs. So these are also involuntary. Okay? Apart from that, what else? Skeletal muscles has got striations. Okay? I'll show you what striations are. These are simply small lines that you are going to see. It looks like this. Okay? What about involuntary? So when talk about the cardiac, the cardiac, this one is also present. Striations also present. When talk about smooth, that's like the name. It's absent. Okay? So here in smooth muscle, there are no striations. That's one thing. And then two, what is the shape? Okay? So the shape of cells, you're going to find that here, the cells are long. So the skeletal muscle cells are going to be long and cylindrical. Okay? Then talk about this. They are short and blanched. So the cardiac, you're going to find that the cardiac muscle cells are going to be what? The shape is short and blanched. It has got branches. Then this one is going to be spindle shaped. Okay. Spindle shaped, what simply means that it's going to look like this. Okay. Yes. So it's going to be what? Small at the end and big at the center. What it means. Then what else do you have to know? The other thing that you have to know, Tim, that will help you identify that this is a skeletal, this is a smooth, this is a cardiac. It is what? The location of the nucleus. The nucleus. So in these ones, the skeletal, you're going to find that there are many, okay, nuclei. Okay, many peripheral nuclei. Now, what does it mean? It means that imagine this is a cell. The nucleus will be what? They're going to be many like that. Okay, maybe one cell has got many nucleus there, and the nucleus are what? On the peripheral, which means they are found at the side, not at the center, but on the sides. Okay, that's what it means. So many nuclei and peripherally located. What about in this cardiac? Usually 1 to 2, and centrally 1 to 2 nuclei and central. They are going to be centrally located. What about the smooth? 1 and central. Okay, so here you're going to find 1 nuclei and centrally located. Yes. What about branches? Okay, skeletal muscle has no branches. What about in this one? The cardiac has branches. Okay, no branches. For smooth, has no branches. Yes. So those branches, we call them what? Intercalated discs. Okay, call them intercalated discs. They are found only in the what? You're going to find them in the cardiac muscles. I'll show you what I'm talking about. Then what else? We also have what we call the sarcomeres. Sarcomeres, these are present in skeletal, present in cardiac, but absent in smooth. So that's why you're seeing that what? These two, skeletal and the cardiac, they have got striations. These striations are due to what? Sarcomeres. Yes, so you have to remember sarcomeres, they make striations. Okay. What else? Regeneration. Just remember that these ones, regeneration is poor. Okay, for the skeletal muscles, regeneration is poor. Yes, but a bit limited. Okay, it's there, but not very good for the skeletal. Okay, for the cardiac, it's very poor, but for the smooth, they're able to generate. So, which means what I simply means to me that once they're damaged, these cells can do what? Can heal, can produce new cells. Okay, so I hope you have some pictures here. so that you can see. Okay, I think you can just use the same diagram. So it's what I was talking about, okay? I'm sure you can see that there are these branches here. Look at the branches, these branches, right? These are what we are calling the intercalated discs. So once you see those things, remember it's a cardiac muscle, okay? What else can you see? Apart from that, we've also talked about the other differences, right? So we've got intercalated discs, this is the cardiac muscle, and then we can also these lines, see these lines, I'm sure you can see the lines, those are called striations. They are due to what? Sarcomeres, right? Yes, and then you can see the nucleus is what? Centrally located. Nucleus central. Let's compare to this. Skeleton, what's the nucleus? Peripheral. Have you seen? The nucleus is on the side. Okay, this one, peripheral. All the nucleus are on the peripheral side. Okay. So imagine this one thing just got many nucleus. Many nuclei. I'm sure you can see that. So skeletal, you find that the nuclei are going to be multiple and also peripherally located. But for the cardiac, it's usually 1 or 2, but centrally located. Okay, I hope we're moving on the same page. And then you look at here, for these ones, the nucleus is what? It's just 1 for the smooth. So you can see the nucleus is just 1 at the center. And remember the cells, how are they shaped? Spindle. Okay, shaped. Spindle shaped, you can also say it what? You can also say that fusiform. When you say fusiform, it's the same thing as spindle, right? It's like this shape. So when you see this shape, just know that this is a smooth. Okay. So those are differences things you have to remember. Let's proceed. So here just simply some few more information on what we're discussing. So skeleton, remember these are attached to bones. Okay, I've talked about these differences. Smooth, they are found in hollow organs, blood vessels, eyes, glands, skin. You find the smooth. Cardiac, you find it in the heart. Okay, now contractile cell functioning are single, okay, cell contractile units. So what are you going to find? You're going to, there's some cells that you're going to find, right, in these in these muscles. So what do you find? We have got what we call the epithelial cells. So these cells, epithelial cells, they simply help in contraction. So in these muscles, you can find some of these cells, as you can see here. Epithelial cells, they help in what? Contraction. 9. We also have what we call myofibroblasts. These are simply fibroblasts that are found in the muscles. They help in wound healing. Okay. Yes. And then you are going to find what we call the myoid cells. 9. Myoid cells, these ones, you mainly find them in the seminiferous tubules. Okay. They are also contractile cells. They just look like smooth muscle cells. Okay. So you find many in the testicular seminiferous tubules. So this also helps they help to propel the sperm. Okay. We also have what we call the pericytes. These ones, they surround the blood vessels. Okay, especially arterioles. That's why you're going to find the pericytes mainly. So these are also contractile units, and sometimes these they're also called what? Ruget cells. That's the other name. Ruget cells. So mainly around the arterioles and capillaries. That's why you're going to find them. Okay. So what you have to remember that these ones they regulate blood flow. Okay. So they can either contract so that the diameter of the capillary should be small, or they can relax so the capillary should be big. That's what the pericytes do. Okay, so those are simply some of the cells you'll find. Now let's talk about the skeletal muscle team. The skeletal muscles. Okay, so what you can see, this is the muscle, skeletal muscle. Okay, you can see this is here. This is a muscle fiber, and that's a muscle fiber. And then there's a nuclei located peripherally. So muscle fibers is the same as cells. When you hear muscle fiber, it's simply muscle cell. Simply another name. So this is a muscle cell. You can see it has got what? Multiple nuclei. They come from what we call myoblasts. Okay? Number many constant usually throughout life. Fine. Yes. So you can see this is a muscle fiber with the nuclei. Okay, so you can see even here, this is the muscle fibers. Okay, now this muscle tissue, it has got connective tissue around it. So what you can see here, this is a muscle. A muscle is connected to a bone through what we call a tendon. Okay, so this is a tendon. So tendon is simply, remember, it connects what? It connects a muscle to a bone. Now this muscle, it has got layers. So do you have a picture? Okay, I think we'll use the same one. It has got layers, right? So it has layers of connective tissue that covers the muscle. So this outer layer, okay, you can see we've got the outer layer there. We've got it, and then these bundles. So what we call the outer layer? So the outer layer, we call it the perimysium, right? So remember, we've got what we call perimysium. This is what the outer layer. Okay, I mean epimysium. Epimysium is the outer layer. Then after the outer layer, we have got the perimysium, and then you have got the endomysium. So remember, the outermost. This is outermost. Epimysium. Okay, so just remember epi, e, okay, for exit, which means outer and outside. Can help you for to remember that. So epimysium, it's the outer. So I'm sure you can see this layer here. This layer. This is what you are calling the epimysium. Okay? And then inside, we have got these bundles. We have got these bundles. Okay? Now, this layer surrounding these bundles, that layer is called what? We call it the perimysium. I'm sure you can see the name. Perimysium. Then now, imagine if you pull one bundle. I'm sure you can pull this bundle in. This bundle here, when you pull one, we call this bundle a fascicle. Okay? So remember, a fascicle is wrapped by what? Perimysium. So a fascicle is covered by perimysium. Right? Then, inside the fascicle, you can see these little round structures. Okay? These. When you get one of these, this one here, this is called what? A muscle fiber. So a muscle fiber is covered by also another connective tissue, which we call endomysium. So I want you to remember that when talking about epimysium, it covers the whole muscle. Then perimysium covers the fascicle, this one. Then endomysium covers the muscle fiber. Now, this epimysium usually is a dense irregular connective tissue. Okay? So I'm sure if you're able to remember what we discussed on connective tissues, this is where we're supposed to apply that information. Okay, so we're supposed to apply that information here. Okay, so anyway, we've got this. So like I was saying, Tim, I was saying that the epimysium is a connective tissue. Okay, and this one is what? Dense irregular connective tissue. Okay. That is the epimysium. What's the function? Protects the whole muscle. That's why it's covering the whole muscle. Yes, and then it continues as a tendon here. When it is here, it continues as a tendon. Then the perimysium, it covers the fascicle, like I said. Okay, a fascicle is simply a bundle of muscle fibers. What is it? It's also a dense, irregular connective tissue. Then the inner one, which covers the muscle fiber, it's what? It's an endomysium. So endomysium, this one is what? It's a loose connective tissue. Loose connective tissue. Okay? So you're supposed to know those things. So you can see, I'm sure this is how a muscle fiber is going to look. So remember, this is the structure. So remember, like I was saying, this is a muscle, it's covered by epimysium. Then here we have got what? A fascicle. A fascicle, okay? So this one here is a fascicle, this one is a fascicle, this is a fascicle. This fascicle is covered by perimysium. And then in the fascicle, we have got these little ones. In the fascicle, we are going to have the muscle fibers. Okay. So if you look at here, this is a muscle fiber. Now, this muscle fiber, it's simply made up of what? These structures here that you can see. What do we call this? We call this what? Sarcomere. So a muscle fiber is simply made up of these contractile units that we call sarcomere. Then the membrane, the cell membrane of a muscle fiber, imagine, because when I say muscle fiber, I'm simply saying what? A muscle cell. Why? It's a cell. So this cell, it's usually like a cylinder. It's round. I'm sure you can see. Like the way it's looking, right? It's like a cylinder. So these cells are cylindrical, and the cell membrane here, we call it what? Sarcolemma. The same way cell membrane in other cells, we call it plasma membrane. Right? Other cells, we call it plasma membrane. The same as cell membrane. But for the muscles, the cell membrane is called sarcolemma. So you have to know those terms. Right? Apart from that, cytoplasm. This is cytoplasm here. For this cell, we call it sarcoplasm. Okay? And then what you can see here, these are simply what? These are simply what we are calling the myofibrils. Yes. Myofibrils is simply, in other words, it's simply like they are made up of what? A lot of sarcomeres. So it's like a bundle of sarcomeres. Okay? So here there are sarcomeres in these, the contractile units. So the bundle is called the myofibrils. And I'm sure you can see these ones here, these lines. This is what makes striations that we see. These striations. Yes, they're made by the sarcomere. So we've talked about this already. Connective tissues. Okay. So when you say fascia is simply a connective tissue to mind. It's a connective tissue that covers a muscle. So muscles usually they're covered outside by what we call fascia. It's another connective tissue that you'll find. Okay, so it's what I was talking about. The same thing. Okay, for epimysia, for these are fascicles. Fascicles, okay, covered. They contain what? They contain the muscle fiber, which is covered by endomysium. Okay, so you can see this is a muscle fiber. Muscle fibers got these myofibrils, which are simply made up of what is sarcomeres. Okay, the sarcomeres, you can see, this is how it's gonna look. It's made up of different proteins. Okay, this is one sarcomere here. So we'll talk about that. Okay. So myofibrils and the structure. So you can see, okay, here, this is what muscle fiber. It has got a lot of myofibrils. If you pull one myofibril, we can see here, okay, simply interconnected interconnected the sarcomeres, made up of sarcomeres. So if you look at here, this is a sarcomere, okay? So sarcomere are interconnected to each other, okay? And you have to know that in the muscles, we have got a lot of mitochondria. So many mitochondria because we want to produce a lot of ATP. Okay, and then if you look at here, you can see that the dark parts here, these dark parts that we see, these are A band. And then the light ones is the I band. Yes, that is I band. Now, I want us to look at the sarcomere team. Let's look at the sarcomere. Let's see, okay. So structure of sarcomere, okay, this is how it's going to look. So the sarcomere, it's made up of these proteins. So we have got different proteins in the sarcomere. So you can see that we have got what we call the actin filament and the myosin filament. Okay. Apart from that, we also have what we call the Z disc. Okay, this is a Z disc. So I want to remember that we have got two types of filaments. So there's what we call the thick and the thin filament. So the thick filament is simply myosin. So this myosin, we call it what? Thick filament. Okay. And then the thin filament is made up of what? Thin filament is made up of actin. So what else is found in the thin filament? So when you hear thin filament, this is the thin filament. We are simply talking about three things, three proteins. Number one is actin. Number two is what we call tropomyosin. Number 3 is what we call troponin. So remember, there are three what? 3 proteins. Okay? 3 proteins. So here what is that's being shown is actin, but there is also troponin and tropomyosin 9, which we'll see later. So if you can look at this picture here. If you zoom at this actin, there is another protein that we call tropomyosin. Okay? And then there is also another protein that we call troponin. So you can see here how they are. So the blue one, this blue protein covering around is called what? Tropomyosin. And then this red one is called the troponin. Okay? And then you have got this purple one, which we are calling what? Actin. Okay? Now, what I want to remember, team, also is that this troponin, it has got what? 3 subunits. Okay? So troponin has the 3 subunits. One is what we call troponin C. This troponin C binds to calcium. Okay? Then number 2 is what we call troponin I. This troponin I is important in stopping what? Stopping muscle contraction. So it's for inhibitory. Then you have what we call troponin T. It binds tropomyosin. So you have to remember that when we talk about the thin filament, we're talking about 3 proteins: troponin, tropomyosin, and actin. When we talk about the thick filament, we're talking about myosin. Okay? Very important too. Then, like I was saying, we have got what we call the Z disc. So, Z disc is simply what? This protein here. Okay? This structure here. This is what we call a Z disc. Now, this Z disc, it's mainly made up of what? It's simply made up of mainly of alpha actinine. So there's a protein called alpha actinine that's inside there. Okay, that's making the Z disc. So we're looking at the structure. Now, the other thing I want us to look at are simply the bands. Okay, so like the way we said, okay, this picture here. I'm sure you can see that because of the way these proteins are, the way the proteins are overlapping, the way these proteins are overlapping, they form these bands. Okay, they are forming these bands. So when we talk about the bands, we have got what we call the A band and also what we call the I band. So I'm sure if you look at here, the A band is simply these what dark lines there, and like I said earlier, the dark lines there. That's what is the A band. So the A band usually is simply made up of what? The overlapping myosin and actin. Yes, so myosin and actin, when they're overlapping, so imagine they say this is actin. Okay? This is myosin. And then here you have got what? This is actin, actin protein. So this is actin, and then this is myosin. So myosin is the one at the center, the one which is big. So this area where they are overlapping here, okay? This area where they are overlapping, it makes what? It looks dark because there are many proteins there. That's why it looks dark. So this is what we are calling what? The A band, okay? Or dark band, okay? Now, then the other area where they are not overlapping, this area here, which is made up of the thin filament. This area here, where they're not overlapping, this one we are calling it what? This area we call it the I band. So remember, I band is mainly made up of what? Thin filament, which is actin filaments. Okay? That's called the I band. I simply stand for what? Light band, iotropic. Okay? So iotropic band. So if you look at here, you can see that this area looks lighter. This is the I band. This area from here to here looks darker, and there are proteins that you can see. I'm sure proteins overlapping. Okay, you can see the proteins. This protein you can see here. This is what the thin filament. Okay, this one is a thick filament here. Thin filament, thick filament. The red one, thick filament. Thin filament. They are overlapping in that area. That's what's making the A band. Okay, so what's a sarcomere? A sarcomere is simply from one I band to the other I band. Right? Yes, in short, you can just say from Z disk to Z disk. I hope people are not lost. If you have a question, feel free to ask. Kind. Feel very free to ask. Okay. So the other thing I would like us to talk about are simply what these bands that you can see, the lines here. Okay. Yeah. So the other bands that you can see here that are important as well, or regions. So I'm sure you can see that you have got what we call the H zone. So this is the H zone 9. So the H zone is this area. Okay, from there to there. Now, what is the H zone? The H zone is simply an area where you only find what? Thick filament, sign, the myosin. Where you find only the myosin. Example where, look at it. This area, we can only find myosin there. So that is called what? H zone. Okay? Now, what is the M line? The M line is simply if you draw a line at the center of the H zone there. It's simply a line passing at the center, okay, of the H zone. So remember, where you find the myosin only, that's what we are calling the H zone. Myosin. That's H zone 9. Yes. So remember, we've got the H zone, okay, the I band, okay, and then we also have what? You also have the M line as well, okay? And then also Z disk. I don't know if there's any question. So what you're seeing here, H band is the same as the H zone. There's no difference there. Okay, so let's proceed. So I'm sure if you look at here now, let's look at these proteins, the myosin filament. You can see this is how the myosin is going to look. It has got these what we call the myosin heads. Okay, so this is a protein. There's also what we call the tail. So it has a tail and the myosin head. These are very important when it comes to muscle contraction. Okay. So you can see this is the tail, and then it has got the head as well there. Okay. Then talk about actin. So you can see the actin protein. It's this one, the dark color. And then on top you are going to have tropomyosin. And also you are going to have what? You're also going to have troponin 9, which has 3 subunits. Troponin T, C, and I, which we talked about already. So troponin has got 3 subunits, which we've already mentioned. Okay. Tropomyosin, what is the purpose? Tropomyosin, it simply covers the active site on the actin. The purpose is simply to cover the actin site where myosin is going to bind. So tropomyosin, remember, it simply covers what? Where it's going to cover the active site on actin where this myosin is supposed to bind. Because remember, we have got the myosin head. This myosin head, it's very important in terms of contraction, the side filament mechanism. Then we also have just these proteins here that you can see. We have got actinin, we have got the other one is nebulin, okay? And we also have titin, desmin, dystrophin. What are the functions? Like, what do they do? So these proteins, remember I told you about alpha actinin. This one makes a Z disk. You find it in the Z disk kind. Yes, that's why mainly you find it. Okay. And then we have got desmin. Desmin, usually, this one, it's actually what connects the myofibrils. So it connects the myofibrils to it together. Yes. So it's not much very important for the exams. And then we have got what we call nebulin. Nebulin, usually this one runs along together with the actin. So it actually moves together with actin. Yeah, so actually this one helps. It's like a molecular ruler. So just remember this one is a molecular ruler. It measures how much actin is contracting or lengthening. So it's able to tell how much. So it actually stabilizes the thin filament. Because imagine if the muscle was just stretching, overstretching, the muscle will end up get damaged. So the muscle has got a limit where it's supposed to end. And it's all thanks to nebulin, because it's able to measure how far. Yes, and then we also have titin. So titin, remember, is a protein that extends from Z disc to M line. Okay, that's how it extends. Okay, along the myosin. So actually, this one, remember, it's the largest protein in the body. Just like the name, why in titan, it comes from the word titan. Titan means very huge. Yes. So you have to, and then dystrophin. This one is very important. Dystrophin, it connects. Okay, it connects the actin filament to the cellular matrix. So it simply connects, very important. Okay, actin filaments to extracellular matrix. Actin filament to extracellular matrix. Okay. Very important. Why? So it's going to help stabilize the muscle cell membrane during contraction. Very, very important. So because the reason I'm talking about this team is that when someone is lacking dystrophin, you are going to find that they develop a condition that we call dystrophy, which we'll talk about. So there is what we call dystrophy, where they lack the sign, the cells get damaged. So remember that this dystrophin, in short, it connects the sarcomere to the cell membrane. So it's keeping the sarcomere in place. Okay, so sarcomere, imagine this is sarcomere. This is sarcomere, and then this is the cell. So this cell membrane there is supposed to attach to. Okay, it's attaching the sarcomere to the cell membrane and stabilizing it. Without this, the cell will get damaged and it develop a condition called dystrophy, where someone become very, very weak. Muscle weakness. That's the feature you're going to find there. So I want us to talk about something now, okay, of the muscle. There is what we call sarcotubular system. Now, what is this? Just a very simple thing, just a name that's sounding complicated. Very simple. Sarcotubular system is simply talking about the tubes. Okay, there are tubes in the muscles. There are tubes in the muscles. Okay, so the tubes mainly formed by two types of structures. So there are two types of tubes, in other words. It's what we call the T tubules and the L tubules. Okay, so the T tubules, okay, or transverse tubules are narrow tubules formed by the invagination of the sarcoplasma. Okay, so we have got these T tubules that you can see. I'm sure you can see these tubules, the yellow ones. These are called T tubules. Okay, and then we also have what we call the L tubules, called the longitudinal, and that's what L standing for. It's standing for longitudinal tubules. Across the tubules that run in long axis of the muscle. Okay, so we are also going to have tubes that runs like this. Okay, so you can see there are these tubules here, and also there are some tubes that runs like this. Are not shown in this picture. So I'm sure you can see. And what is it saying? They are made by what? The cell membrane. Imagine this is a cell membrane. It just go in. The cell membrane goes in and makes the tube like that. So this is the sarcoplasma. Okay, or cell membrane of muscles. Makes these invaginations. These are the tubules. Very important tubules. Because when we talk about contraction, these are important. So the T tubules, what's the function? They're important for rapid transmission of impulse. Okay, in the form of action potential from sarcolemma, which is the cell membrane, okay, to the myofibrils. The L tubules, what's the function? store a large quantity of calcium ions. When action potential reaches the cystine of L tubule, the calcium ions are released into the sarcoplasm. Okay. Now let's talk about the mechanism of what? Muscle contraction. Okay. So mechanism of muscle contraction, there's what we call the slide mechanism, slide filament mechanism. Okay. So when you talk about muscle contraction, team, the first thing you have to understand is that there's going to be action potential that comes in. Let me just add the page. So the first thing is the action potential. Yes. So action potential is coming from a cell. So imagine you're coming from a neuron. So you're going to have a neuron that is sending action potential. Okay. And then this action, let me just draw that. Okay. So imagine, let's say this is a neuron. Okay. This neuron, it's bringing what? Action potential. So action potential will come in a neuron. And then when the neuron reaches here, this action potential, it's going to allow what? The influx of calcium ion. So action potential activates what we call the voltage-gated calcium channels. Voltage-gated calcium channels. Okay. So these calcium channels, when they detect voltage, action potential, they open. And once they open, what's going to happen? When they open, calcium will enter. Now, why calcium entering? Because in this in this neuron, there is what? There are these vesicles. Okay. These vesicles, they contain what? Acetylcholine. So the purpose is that there's going to be what? This calcium, it causes these vesicles to come and fuse with the cell membrane. So I want to remember that the cell membrane here, this is the cell membrane of a neuron. There are proteins on the cell membrane there. There are some proteins. Okay. There are some proteins. Okay. So those proteins there. And apart from that, we also have proteins here on the vesicle. Yes. So there are different proteins. Some proteins are on the vesicle, other proteins are on the cell membrane. Okay? So these proteins, what happens that they start interacting. And the calcium, it comes and helps these proteins to interact like this. That's the purpose of calcium. So remember, calcium helps proteins to interact, and then we are going to pull this vesicle toward here, toward the cell membrane. And then we are going to release through what? Exocytosis. Okay, so remember that the purpose of calcium is that it activates these proteins. And so I've got what we call syntaxin, we've got what we call synaptobrevin. Those are simply some of the proteins. So these proteins, they interact together, and then they cause this acetylcholine that is there to come out. That's a simple thing. So we are going to release calcium, in short. Acetylcholine. Acetylcholine. So the purpose of action potential is simply to help us release acetylcholine. Now this acetylcholine, when it's released, there is what? There is a muscle here. Okay? So there's going to be a muscle there. So this muscle, this area, you are going to find that there are going to be what? Different. There is what? There are proteins here. Let me draw there. So there are going to be proteins there. Okay? So this protein here that you can see, it's called what? Nicotinic. Okay? Acetylcholine receptor. So you have what we call a nicotinic acetylcholine receptor. It's simply a protein that is there. This protein, it allows acetylcholine to bind. So acetylcholine, when it's produced, it will come and bind to the nicotinic acetylcholine receptor. Okay? When it binds to this channel, it allows sodium to enter. So we can say it's a ligand-gated sodium channel. What it simply means when you say ligand-gated sodium channel, it simply means that when something binds to it, a ligand binds to it, it opens and allows sodium to enter. So remember, there's going to be a protein called nicotinic acetylcholine receptide, which allows sodium to enter. When sodium enters, it causes action potential. So action potential start moving now in this direction. Okay? We'll move like that. And then this is what we are calling the T tubules. They are going to be like that. So this action potential will keep coming, and then you start going down the T tubules like that. Okay? So it will keep going down the T tubules. Yes. So you have to remember, it will keep going down the T tubules. Now, one thing I want you to remember, colleagues, again, is that in the T tubules here, there are these channels. Okay? There are these protein, these receptors. That receptor there. And then you are also going to have these structures here. There are also these structures here. This is a cell. This is a muscle cell. This is a muscle cell. In the muscle cell, we're also going to have this structure. What do we call this? This is the endoplasmic reticulum. Endoplasmic reticulum. But here we call it what? Sarco. Remember I said the name changes for the muscle cells. Endoplasmic reticulum is called sarcoplasmic reticulum here. So what's going to happen, colleagues, is that in the T tubules, okay, in the T tubules, there are these proteins. Okay. What do we call this protein? This protein, we call it dihydropyridine receptor. Okay, dihydropyridine receptine. That's what we call it. Okay, it's found in the T tubules. This is very important, especially when you're explaining your muscle physiology. This one. And then here, on the endoplasmic reticulum or sarcoplasmic reticulum, we also have another protein there. This protein is attached to the dihydropyridine. Okay, they are attached, they are connected like that. Okay. Now, what do we call this protein, which is found on the sarcoplasmic reticulum? That's why many people get confused. It's called what? Lyanodine. So don't confuse these two. Lyanodine receptor. So what happens to me is that action potential, when it's coming, which is simply positive charges, when they are coming, these positive charges, what do? They activate this dihydropyridine, which also activates the lyanodine receptor. When you activate the lyanodine receptor, what happens? This sarcoplasmic reticulum, it's a storage of calcium. Okay? It's a storage of calcium. Okay? So this calcium, once it's activated, this lyanodine receptor team, calcium will come out. Calcium comes out. This calcium, when it comes out, where does it go? It goes to the troponin. Okay? So remember that we talked about the proteins, and we said we are going to have what? The actin. Okay? We have actin protein like that. And then you also have what we call the troponin. Troponin. This troponin has got what? Troponin C, troponin T. So calcium comes and binds to troponin. Like that. To troponin what? Troponin C. Now this troponin C, once it binds to calcium, it changes the shape. Now remember I also talked about another one molecule that we call what? Tropomyosin. Tropomyosin, we said, it covers like this. It covers what? It covers the active site of what? Of actin. So tropomyosin, what I want you to remember, imagine team, is that imagine, let's say this is actin. This is actin here. Now actin has got a what? An active site. This active site, the red one, which binds to what? Which can bind to this, which is myosin. So actin has a site which can bind to myosin, but we don't want actin to bind to myosin. So what happens that we pass what? There's this molecule that passes there, which is called tropomyosin. So remember, tropomyosin simply covers the active site on actin, so that myosin should not bind to actin. That's the purpose of tropomyosin. Now here we want action potential, meaning we want muscle contraction to take place. So what's going to happen is that this troponin, when it become activated, it's going to move. Okay, it moves what? Tropomyosin from the active site of actin. So we are going to displace the tropomyosin away. So remember, calcium binds to troponin C, it becomes activated, and then it moves or displaces tropomyosin, okay, from the binding site, okay, on actin. Yes. So once it displaces that, then the other step now that's going to happen, team, is that we are going to have what we call a cross bridge formation. Cross bridge formation. What does it mean? It simply means that myosin is going to bind now. Okay, we have a picture here anyway. Sure you can see. Let's go. Let's see this. I'm sure you can see what's happening here. So now after that, you can see that now this is the myosin. Myosin has got head and the tail, like I said. So myosin now binds to what? To actin. Since tropomyosin, which was covering, has moved away, myosin will bind to actin. And that's what we call the cross bridge. Okay? So you have to remember that. So now, which one binds to this? The one that binds to the head, the myosin head which binds, it's the one that has got what? That has broken down ATP. So for this myosin to bind to actin, it's supposed to break ATP in. So once you break down ATP, the energy that you are going to have, the myosin head, the energy that it gets, is what helps it bind to actin. This is why ATP is used in the body. Very important time. So remember, the cross bridge to occur is what we call a cross bridge. For it to occur, myosin head, first ATP binds to myosin head. Then ATP is broken down into ADP and the phosphate. Then the myosin head binds to the binding site on actin and forms a cross bridge. Okay, so you can see there. Okay, myosin head binding there, ADP there, you can see, so it's binding in there. So the first thing, it goes, ATP is broken down to ADP and phosphate, then it attaches nicely. Okay, yes. Then the other thing that I want to know is that once it attaches, is what we call now a power stroke. Okay, so power stroke, what happens, team, is that during a power stroke, it means that, look at what happened, team. The myosin head, look where is it? The myosin head is like this, it's facing here. Look where it is, it's like this. So what happens is that when this myosin head is going to move like this, forward, when it moves forward, this actin also moves forward. Then that's what we call power stroke. Okay, so remember, the myosin head, it moves towards the M line. So it's going like this. Remember the M line, we said it's a center. So this myosin head is going to push the actin toward the center. Okay, that happens during what? We are going to release ADP. When you release ADP, that's when it happens. So I want you to remember very well, team, that the first thing, myosin head. Okay, ATP binds. Second, this ATP, okay, is broken down ADP into ADP plus phosphate. Then it binds to what? It will bind to actin. That's what we call a cross bridge. Okay. Then what happens? This ADP, okay, this ADP, we release it, and phosphate are released. What happens once they are released? This guy, the myosin head, it pushes the what? The actin toward the midline. This is what we call power stroke. So you have to remember that time. So power stroke is simply now ADP now has been released. Then after that, what happens? Another ATP will come. ATP will come and bind again. Then this guy now, this myosin head is going to leave what? The actin alone. So it means it's going to detach. So it detaches when another ATP binds in. Yes. So remember, without ATP, myosin cannot detach from actin. Okay? And it's going to produce what? Rigidity now. This is what we call in what? This is what we see in rigor mortis sign. What is rigor mortis? Rigor mortis is simply what the state in. The state when someone is dead. When someone is dead, especially during like mortuary, okay? I'm sure we are able to see like the way the body looks in. You find that when someone is dead, the body is going to be stiff. Body is going to be rigid. Especially within what? The first 12 hours. The body will be very stiff. That's what we call rigor mortis. Okay? Now, this rigor mortis, how does it come about? It's coming about because if a person is dead, there's no ATP being produced. So without ATP being produced, the myosin aid, it remains binding to what? To actin. It cannot detach. For it to detach, are you able to hear me, Tim? Am I audible? After I thought I'm alone in the meeting, eh? Okay.
Yes, sir. We can hear you, sir.
Okay, madam. Okay. Okay, yeah. So what I was saying that rigor mortis, it's simply what? It's simply like the state of the body when a person has died, where the body remains stiff and the body is rigid. This actually helps, especially in forensic sciences, for estimating the time someone has died, the time that has passed. That's what they use. So this rigor mortis, I was saying, what happens is simply what? There's no ATP, so the myosin aids, they remain attached. They remain attached to actin, because for them to detach we require ATP. Okay, so they are going to remain locked. That's the key thing that you have to remember about rigor mortis. Okay, so basically you're supposed to remember that time. So that's the same cycle. That's what's happening. Then how does muscle relaxation comes about? Muscle relaxation comes about when a nerve now stops producing acetylcholine, the stimulant. Okay, because it said muscles is excited. Why? Excitability. Muscles are excitable. So now once you stop producing the stimulant which is exciting the muscle, that's when the muscle is going to relax. Yes. So remember that when you stop producing acetylcholine, what happens? There's no action potential there. Yes. And by the way, I want to remember that this area here, this junction between a neuron and the muscle is what we are calling what? Neuromuscular junction. We call it this neuromuscular junction. That's what we call this area. Okay, we'll do nicely this topic again in physiology. And then this area where this side of the muscle, the side of the muscle that is facing the neuron. I'm sure you can see this is the neuron, but the side of the muscle that is facing the neuron, we call it a motor end plate. Okay, motor end plate. And simply side that's facing the neuron. Yeah, so what I was saying, Tim, is that what happens, how do muscle relax? For muscle to relax, acetylcholine should go down. No acetylcholine, no action potential. If there's no action potential, what happens? Here, you're supposed to remember that there are what? There are channels they are in. There is a channel. Okay. So there's a pump actually. It's a pump. So the pump is found there on the sarcoplasmic reticulum. What do we call it? We call it a SECA. SECA, what does it stand for? It simply stands for what? Sarcoplasmic. Okay. Sarcoplasmic stroke endoplasmic. Okay. Reticulum. Okay. Calcium. ATPase. So what it simply means, fine. So that is called a SECA. So a SECA, sarcoplasmic stroke endoplasmic, reticulum, calcium, ATPase. This is simply going to pump the calcium back into the sarcoplasmic reticulum. When you pump calcium, what happens? Troponin C has nothing. Okay? Troponin C won't have what? Won't have calcium, like we said. Then troponin C has no calcium. Okay? So what happens? Tropomyosin will go back and cover the binding site. Okay. Tropomyosin covers the binding site and the muscle returns to its resting state. So that's contraction team. So what we are calling the sliding filament chain, because it's called sliding filament because the actin slides over myosin. That's why we call it that way. So this is the same thing. Okay. So sarcomere is going to shorten. When the muscle contracts, sarcomere shortens, as you can see, and then it's going to do what? Again, it expands. Okay. So this is a relaxed muscle, this is a contracted muscle. You can see the difference. Okay. And these are simply stents. Yeah, and then I think I want you to remember, team, that muscles, remember these skeletal muscles, they generate time. Yes, so cells that form skeletal muscles are called myoblasts. And then we have what we call satellite cells. Satellite cells, these they serve as potential source. So for example, let's say your muscle is damaged. If the muscle is damaged, some cells can be produced, okay, and can be replaced back. Okay. I'm sure you're able to remember those tissues that we discussed last time. The labile, the permanent tissues, all those, they are coming in here. Yes. Which tissue does the skeletal muscle fall under? Anyone who can remember? Which tissue? Is it labile permanent? So this one classify them as the permanent. Yes, many as permanent because the level of regeneration is not good. Okay, so they can generate, but mainly these ones generation is not good, right? As compared to epithelial cells. Okay, so remember I've got the stable. Remember we said the stable is the liver, the kidneys, fibroblasts. Those are stable. Smooth muscle cells are stable. What do you mean? The stable normally they don't divide, but once they are injured, that's when they start dividing. Same applies to the liver. When you cut half of the liver, it can grow back because the liver is able to regenerate in the liver cells. Permanent tissues, these ones do not divide. Okay, they have little to no ability. That's what you have to know. Little to no ability. Right? So, for example, skeletal, the neurons, okay, cardiac. That's why when the heart muscle is dead, so of course it's gone. Okay, neurons. So many of the neurons, the cardiac and the skeletal, these are falling under permanent. Then the labile, these are continuous dividing throughout life. Skin epidermis, and the epithelial cells in short. Epithelial cells, okay, bone marrow cells, all those. Okay, let's talk about smooth muscle quickly, team. So smooth muscle, you have to remember that this is also another type of muscles that we talked about. The cells are actually going to be spindle shaped. You can see 30 micrometer in length, non-striated, involuntary, supplied by autonomic nervous system. Why? Autonomic nervous system, team, is the one that we cannot control. Okay, for example, the sympathetic and parasympathetic, those are fall under autonomic, which means that you have no control over the sympathetic. So sympathetic nervous system is simply a group of nerves, okay, that you cannot control. For example, the nerves that goes to the heart. Okay, they have got sympathetic and parasympathetic kind. Yes, myofibrils and sarcomeres are present. Okay, so those are the features. Now, when talk about these cells, remember we have got what the sarcomeres are present. Everything that we discussed, actin filament is going to be there. Thin myosin filament will be there. Okay, Z discs the same way like the way we discussed in skeletal muscles. Yes. Now, I want to know that we've got types. We've got what we call the visceral or unitary smooth muscles. Okay. So these ones they contract as a functional unit. So imagine let's say I've got smooth muscles. Okay. Imagine you say these are smooth muscles. These smooth muscles, you have smooth muscle there, and you also have another smooth muscle here. These smooth muscles, when one contract, this one contract, they all contract at once. Yes. So it's when you say single unit, okay, or visceral or unitary, it's the same thing. These are simply smooth muscles that contract together simultaneously. Why? Because they have got gap junctions in between. Gap junction, these are simple junctions that allow electrical impulse to move quickly. Okay, so imagine this action potential. It starts in this cell 1, quickly to pass through this gap junction and go to cell 2. So both cells are going to contract almost at the same time. So remember, functions as a unit, they work as one, these muscles. So you find these visceral or unitary smooth muscles, you find them in the intestines, in the stomach, because for example, let's say stomach, stomach, it can be bad. Imagine the stomach, one side is contracts, then the other side is not contracting. No, we want the stomach, when it contracts, all of it is contracting. Okay, so those muscles are supposed to work together as one. Same applies to the uterus, uterus, bladder, ureter, even intestines. Imagine one intestine, half is contracting, the other half is not contracting. We can't push food. So we need what? All of them to contract at once. Those are called the visceral or unitary. We also have what we call multi-unity. Okay, so these ones, it means that each cell, imagine this cell here, this cell there, this cell there. Each cell is what? Has its own nerve. Each cell has its own nerve. Okay, so it means each cell is contracting at its own time, not together. So where do you find it? Usually find this what? You're going to find it mainly in the iris of the eye. Okay, you find it also in the large airways, like the trachea, you find that. Okay. Yes, so the smooth muscle is actually located in many areas. Okay, so you can see the small intestine, large intestine, blood vessels. Okay, you can see in the ureter. They are located in so many tissues. Okay. Functional properties of smooth muscles. Some of the smooth muscles exhibit what? Autorhythmic contraction, which means they exhibit contraction at the same time. Why? That's what it means. And also they have got a rhythm
I don't know if it's from my end. I can't get you, sir. Even me, I can't get him. I've just seen the slides moving.
Okay, are you able to hear me, Tim? Sorry, I think in network. Am I audible?
Yes, we can hear you now.
Okay. Yeah, so like I was saying here, what happens, Tim, is that when look at the mechanism why in contraction in smooth muscles, like I was saying, first calcium enters and it combines with calmodulin. Okay, that's what I was saying. Calcium enters and it combines with calmodulin. Calcium can come from outside and also from inside, from sarcoplasmic reticulum or from outside. That's how calcium enters in the smooth muscle cells. So calcium enters, binds to calmodulin. I said smooth muscle cells do not have troponin. They have what we call calmodulin. So calcium and calmodulin, when they combine, they form what we call calcium calmodulin complex. Calcium calmodulin complex, it activates an enzyme that we call the myosin light chain kinase. Myosin light chain kinase, what does it do? It's going to phosphorylate myosin. Okay? When you phosphorylate myosin, what's going to happen? You're going to activate the myosin ATPase. And then myosin will bind to actin. Okay? So just remember that when you phosphorylate myosin, it simply means that we are adding phosphate to myosin aid. And then once we add phosphate to myosin aid, okay, there's an enzyme in myosin aid that we call myosin ATPase. That enzyme becomes active and muscular contraction is going to take place. Okay, so this is what we call the latch bridge mechanism. Okay, we call it the latch bridge mechanism. It's simply the same way we're calling stride filament mechanism there. Okay, this one we call it latch bridge mechanism. Right? We'll discuss this in more details in physiology. Okay, yeah, so just remember that. So control of smooth muscles, like I was saying, smooth muscles can be controlled by nervous factors, right? Sympathetic and parasympathetic nerves. They can control smooth muscles, which means they can activate contraction. Humoral factors, right? For example, hormones. Hormones such as what? Hormones like oxytocin. So hormones can activate time. For example, like oxytocin and angiotensin 2 can activate that. Okay? So these are also neurotransmitters like acetylcholine, ADH, adrenaline can activate that time. Even things like what? Lack of oxygen, excess carbon dioxide, these can cause relaxation. Okay? Lactic acid, potassium, too much of potassium, low calcium, they can cause relaxation. So remember, they are simply something that can control the smooth muscles. Yes. Cardiac muscles also, this is found in the heart, as we know. Okay. So many similar structural and functional characteristics of skeletal and smooth muscle cells. They exhibit what is striations. They have shorter muscle fibers. They have branches, like the one we said at the beginning, 1 or 2 nuclei placed centrally. They are also involuntary. Okay? And they contract automatically. You don't have to contract them. They contract automatically. So you can see this is how it looks. So if you see something like this, you can see there are spaces in between, and there are also these branches. Remember, this is this cardiac. So these branches are called what? Intercalated disc. Very, very important. Okay? Usually in the exam, they can bring a picture and then tell you label or what is this type of muscle. So remember, cardiac works as a syncytium. Okay, what does it mean? It means that it works as a whole unit. So one muscle contracts at the same time. Okay, so the heart muscle contracts at the same time. Right? That's what you say, functional syncytium. Okay, it simply means that the muscle contract at the same time. Why? Because they are what? These branches, they are these branches that we are calling these branches. We say that these branches are called what? They are called intercalated discs. Now, in these discs here, there is what? In these junctions, there is simply gap junctions. So there are little gap junctions there. That the same way I was talking about smooth muscles, even here there are gap junctions. That helps what? The ions and the electric impulse to move quickly from one cell to the other. Okay, that's what you have to remember. So The cell junctions contains gap junctions and also desmosomes. Yes. So these they help simply move electrical impulse from one cell to the other as quickly as possible. That's why these cells contract at the same time. Okay, and we call them syncytium. Okay, then we also have what we call the Purkinje fibers. Okay, which are part of the conduction fibers. So remember that in the heart we have got these like I can say they are nerves. Okay, basically they are like nerves. But in reality they are simply what modified cardiac fibers. Okay, so they are modified cardiac muscles, but they look like nerves. So these they make the conducting system of the heart. So remember we have what we call the Purkinje fibers. Okay, so this is the Purkinje fibers, how it's going to look. Okay, so it's simply a modified cardiac muscle that helps transmit the impulse. Okay, so you have to remember Purkinje fibers, they are very important part of conducting system of the heart, and we'll discuss this when we deal conducting system of the heart. 4 properties of cardiac cells. So you have to remember we have got excitability. Okay, we have got conductivity, and then we also have contractility, and we have rhythmicity. So excitability, it's simple. Simply means that it's able to do what? To respond to stimulus. Okay. Conductivity, it means that it's able to transmit electrical impulse from one cardiac cell to another through the gap junctions. Okay, in the intercalated discs. Okay. Then we have got contractility. This is also called what? Ionotropy. Ionotropy. That's another name. Ionotropy. Ionotropy simply means that it's simply talking about the ability of cardiac muscles to contract and generate force after being stimulated. Okay? Yes. And then rhythmicity. It's able to generate what? Rhythm. Okay? It follows a particular pattern when it's beating. Okay? Or when it's contracting. Okay, so this is how the cardiac muscle is going to look like. It has got all those structures that you can see. Okay. Okay, so these are simply some of the markers, not very much important. Now, that's the last part, Tim, that we can talk about is muscular dystrophy. Yes, like I said, it's a condition where someone is going to lack what? The dystrophin protein, okay, which holds the sarcomere. So I'm sure you can see here, this is a dystrophin protein. It's holding what? It's holding, so all this structure here is a complex. So it's holding what? It's holding the actin to the matrix, okay, on the cell membrane later on. So remember that now people, they can lack what? They can lack this protein, okay? When they're lacking this protein, they are going to develop what we call dystrophin, okay, muscular dystrophy. So this muscular dystrophy is a condition where someone is lacking this. Now, there are 2 types of muscular dystrophy, Duchenne and Becker, okay? So what you have to know is that when talk about these 2, in Duchenne, it's more severe. So Duchenne is more severe. And what's happening? You have to remember that in Duchenne, this one is most common type. And what you have to know is that dystrophin is actually absent. That's why it's more severe. So dystrophin is what? Absent. Completely absent. This protein. But when we talk about Becker, dystrophin is there, but it's reduced. Okay? It's going to be what? Reduced. That's why this Duchenne, since there is no dystrophin, you're going to see the symptoms early, usually around 1 to 5 years. The symptoms are going to show. Someone is going to start having what? Become very weak. Okay? You're going to remember that this is what Duchenne. So it's actually very severe. And then talk about Becker. This one usually started during late adulthood, or maybe sometimes adolescence. That's when it's going to start. So you have to remember there are 2 types. So remember Duchenne, most common caused by defect. Okay, that makes a protein dystrophin. Without the protein, the muscle breaks down and person gradually becomes weaker. Symptoms: wasting of muscles, poor balance, limited range of movement. Sign is so you can say dystrophin there. If it's not there, you can't hold the sarcomere. Okay, so Duchenne, simply some of the features that you can see. Okay, then Becker, this one, like I said, it's the milder version 9. So the protein is being produced, but the protein is not produced as the normal. Okay, so there's going to be progressive muscle weakness, right, which usually start around adolescence, sometimes late adulthood. Okay, so these are simply some of the features, and now you can diagnose it. Okay, so these are simply the types you can see there. The other condition we can talk about is simply myasthenia gravis. So myasthenia gravis, this is simply also a condition that affects the neuromuscular junction 9. So it's an autoimmune disease. So imagine you've got a condition that where the cells, this here, I'm sure you can see, I mean, these proteins, the acetylcholine protein 9, acetylcholine receptor, they are, let's say you've got IgG antibody. Your body produces the antibodies against this. So it's like your immune system is attacking the acetylcholine receptor. So these acetylcholine receptors get damaged. When they get damaged, it means what? Acetylcholine has nowhere to bind. So you can see here, this little green one. These are the IgG antibodies. They bind to acetylcholine receptor. Acetylcholine has nowhere to bind. So which means there's no contraction. Someone, even if you say you want to move, you want to walk, of course, you can't contract the muscles because the acetylcholine receptors, some are damaged, some are occupied with the antibodies. So it developed this condition, which is called what? Myasthenia gravis. And most of the things that you are going to see, we are going to see ptosis. Okay, this is what we call ptosis. Ptosis is simply drooping of eyelids. Yes. Apart from that, you also see mainly diplopia. Diplopia is simply double vision. Okay, and then sometimes also affects the muscles of chewing. So someone is going to have difficult chewing, someone is going to have difficulty in swallowing, which we call dysphagia. Okay, so you see difficulty in swallowing, fatigue, okay, weakness of the neck, all those things. That's what we call myasthenia gravis. Okay. I don't know if there's any question, team. Otherwise, we've covered a lot. Any question? Okay, I think if there are no question, I think we are going to call it a night. We'll end here. Then we're going to meet you on Saturday. I mean, on Sunday. Our next meeting is going to be on Sunday. Yes. And then now we'll go for residentials. After that, that's when we'll come and continue again. Okay, team. Have a blessed night.
Thank you.